to get us started today by introducing Riley for her prelim seminar. So she started out as an undergrad at Lincoln Memorial University on a lacrosse scholarship. Fun fact, you may not have known about her. And then she ended up transferring to Clemson, which we're very glad about because it got her started on the path that led to her being here. So she got very involved in microbiology research as an undergrad, including doing a lot of work on microbiomes in herps, including lizards. And so we've sort of been like dancing around working on the microbiome, and I'm super excited that Riley is like really taking this on. And so her interests broadly are in host-associated microbiomes and in the different factors that structure them, which is what she's going to talk to us about today. Yeah.
So thank you everyone for coming. I really appreciate the turnout. It's gonna be so much fun. A quick disclaimer about photo credit. So if there's not a photo credit on the slide, that means the credit goes to the Daphnia lab. And that's gonna be the majority of the photos I showed today. So let's get started. Like what Meg alluded to, I'll be discussing the potential role of biotic stressors, commonly occurring biotic stressors, in shaping host-associated microbiomes of Daphnia, which are a zooplankton. So look at the screen, pretend that you don't know what this blob is supposed to be, right? You have no idea what it's standing for. However, right now, we can think of it as a host. Now, a host is simply an organism. It can be macro or micro, which occupies an ecological niche where it consumes resources, interacts with other members of different species or even members of its own species, and similarly reproduces. So what is the defining role of the host then? Now, it is serving as the greater environment for symbionts. And now symbionts are smaller organisms that live in close association with the host. And now this association is highly variable regarding the relationship type between the host and symbiont. So if you take a peek at that top corner here, you can see a gradient and it's a little vague. So we have mutualism, and this is when both the host and the symbionts receive a benefit in this relationship. A common example are nitrogen fixers and the rhizosphere that can provide benefits to their greater plant host, which in return provides some sort of carbohydrate reward, right? In this case, we can see these gut symbionts here probably helping with some sort of nutrient assimilation. In return, they get a piece of that pie, right? So both parties benefit. Now, if you go to the opposite end of the spectrum, you have parasitism. Now, this is when you have your symbionts taking advantage of the host. They steal resources, they steal ATP, and this is at the detriment of the host organism and can lead to eventually host death, right? So I know what you're all thinking right now. Is this all happening at the same time? Okay, unfortunately, yes. So we can do this really quick literature search where we can see that there's actually this tripartite interaction occurring between the host, the microbiome, and the parasite. And the perspective most people take is that the microbiome mediates the host-parasite interaction. And I want to take a quick look at this paper right here by Kayla King, which in the C. elegans model, they showed that a mildly virulent pathogen actually switched over to a more mutualistic lifestyle strategy in the presence of this novel, more virulent pathogen, and therefore conferring this resistance against the parasite to its host, right? Now, we're operating in a pretty specific system at this point. So as you can probably deduce from this blog, it is actually a Daphnia, surprise, surprise, right? And so let's think about this interaction in the context of my system, the Daphnia system. So let's remove this bidirectional arrow here and actually reconsider the role of the microbiome in parasite-host interactions. And what we find is there's either no role or a poorly understood role going on here. So we do not find the microbiome conferring any sort of resistance against parasite infections. They don't really have this elucidated antagonistic response against potential invaders. However, this is a unidirectional relationship as the parasite can actually modulate the microbiome and therefore infect the host through the microbiome. And we see this in some studies that show that in the field, a gut parasite is able to disassociate the original microbiome and have it diverge. And this is caused by this high microbial community turnover. So you have these distinct microbiome profiles between infected and uninfected individuals. So let's take a little bit of a closer look right here. So we're going to be focusing mostly on the gut microbiome today. This is a conserved region in the Daphnia, where you have like this inflow and this outflow. And if you can take a closer look, if we look at these gut epithelial cells, there's this consortia of diverse microbiota living in close association. So, what happens when we introduce a stressor such as parasite exposure? So you have these big bad spores up here, and they are definitely doing something, right? So this can go broadly in two directions. First, we're going to discuss dysbiosis. Now, dysbiosis is characterized by a partial or complete turnover in a core microbiota, and that core is defined by a threshold of your choosing. And so what happens here is that the parasite spores interact and they cause a reduction in the alpha diversity of the community, like present in there. This is really a stochastic shift. There's no determined way, there's no adaptive response. However, there's a potential for an adaptive microbiome to occur where there's a directional shift that is specific to the stressor, in which those sensitive microbiota die off, and those that were already present encoding some sort of beneficial response against this parasite begin to proliferate under the new stressor, right? And this can lead to a happier host. So let's talk more about Daphnia as my specific host system. So these guys are a model organism in genetic, ecological, and evolutionary studies. They are a key grazer in freshwater ecosystems. This means that they consume a ton of algae, and they also serve as an important food source for fishes. They are predominantly asexual, which is really important in the lab as we can have these genetically identical clonal lines set up. Short generation time, so you can go through them rapidly. And importantly, Daphnia are transparent. Now, this allows for a quick visual diagnostic of the many parasites that these guys can harbor. So in my talk today, what we're going to do is first distinguish between the direct effects of parasite virulence and then the indirect effects of microbiome dysbiosis on host fitness. Moving forward, I'm going to propose a way to test for a potential additive or synergistic effect of two common biotic stressors in reducing host fitness as mediated through that host-associated microbiome. And once I'm informed by these first two chapters, I will then take this out into the field to investigate microbiome reassemblage under both stressors in the natural environment. So let's go back to that first question. What I'm asking here are patterns of disruption the same across different parasite species? And what I mean by that is when you have these different parasites unique in their mode of infection and virulence, are they causing the same sort of changes in community composition of the microbiome and then the subsequent functional capabilities, right? So why does this matter in the first place? Well, first of all, the direct and even indirect consequences of parasitism on Daphnia microbiome function is basically unknown. There are potential effects for this later affecting the host's greater role within the ecosystem, hypothetically maybe increasing like a chance of population crash. And because there's such a high diversity and prevalence of parasites in these natural systems, it would be nice to know, do they all lead to the same generalized scaled up effect? So thus to gain a better understanding of the overall response of the host and associated microbiome to parasites, I will experimentally infect my host with different parasites and then quantify the shifts in microbiome community composition over time. Okay, I'm going to take a sip of water because I'm going kind of fast. So we know that Daphnia rely on their microbiota to survive. What this study did was produce a xenic Daphnia and then monitor their lifespan, right, and other host fitness traits. And what they found is that Daphnia die after 2 weeks if you keep them germ-free. They also don't reproduce and their body size was greatly reduced. In addition, once again, we know that infection causes this new community composition to occur distinct from that original healthy microbiome. So if we take a look over here, all these dots signify as a pooled sample of Daphnia guts, and the red is for this parasite infected sample, and this is for uninfected. And so we can see that these are clustering independent of one another, having these distinct community profiles across different diversity metrics. And then finally, let's kind of revisit the idea of what is the microbiome even doing in these parasite-host interactions? And the answer is we are unsure. So from this study, they, across the same genotype, they cultivated these azenic Daphnia and hit them with 3 different microbiome inoculations, and then saw if there was like a difference upon parasite re-exposure using body size as a proxy. And there was no really significant difference. They were about the same effect. So let's think about this closer to home and data collected in our lakes. So in the 2021 field season, the Duffy lab collected and sequenced Daphnia from North Lake and Sullivan, and these were processed by collaborator Ray Cooper. And what they looked at was the relative abundance of certain phylum in unaffected and infected hosts. And you can see this overabundance of firmicutes in infected hosts versus the uninfected hosts. So yes, again, distinct microbiomes. What are they actually doing, right? So some quick limitations here. The samples were not controlled for the presence of different parasites within the host, or if there were any artifacts left over from the way the samples were collected. And there was also no causal evidence applied that these differences in the microbiome relative abundance was even driven by infection, or whether or not this shift translates to lower host fitness in the field. Thus, I'm going to try and experimentally isolate these effects in my chapter 1. So let's get familiarized with the parasites I'll be working with. So I'm going to, for brevity's sake, just call these guys by their nicknames. This is Mech, right? Mech is a highly virulent yeast parasite, and how it enters the host is via filtering, so it goes in through the gut. And these guys have needle-like points on either end, hence their name by Pipsidata. And once they're in the gut, they're able to puncture that gut wall and spill out into the host hemolymph, where they then reproduce. Once again, these guys kill their host pretty quickly, within 2 weeks normally. And they will lyse the body, spreading thousands of spores out into the water column. And you can see a quick here of some mesh growth, and this is an uninfected Daphnia. So my next parasite is a microsporidium parasite that similarly interacts with the gut as Mech does. It is an obligate intercellular parasite. This means it accumulates within the gut epithelial cells, where it then begins to proliferate. And as it causes that cell to grow, it eventually shreds its microvilli and eventually lyses. However, MICG has a low virulence on the individual scale, and so their lifespan is not overly affected. And you can see these little white clusters, oh my goodness, my hand, these little white clusters in that gut, right? This is an infected individual. Now, our final parasite is one that, as far as we know, does not directly interact with the host gut. So it's going to serve as a kind of loose control. It is an oomycete parasite, and what it does is that instead of the gut, it infects the host's brood chamber where it consumes the host's embryos, but thus lowering fecundity, but not necessarily lifespan. So to kind of go back, as I spoke a lot, this is the question. We're looking at all these super diverse parasites, and I'm wondering if they're causing the same effects, right? So some predictions to guide us through the methodology that I'll get into is that I expect my parasites that directly interact with the host gut, so Mech and MICG, to have a greater magnitude of change in microbiome composition from beginning to end of infection. In addition, I expect to see a decrease in microbial abundance and biomass as infection proceeds in all my parasite treatment groups. So I'm expecting that Mech will cause this greatest decrease because it causes this traumatic injury within the gut wall that can cause a loss of potential colonization space for symbionts. And lastly, once again, alpha diversity should lower, and I should see higher community turnover across my treatment groups and infected and uninfected individuals. So in order to determine how the microbiome structure and the gene expression changes across infection status, let's set up our treatment groups. So genotype actually holds a lot of influence determining the microbiome composition. And so to mitigate these differences, I'll be using a single clonal line of Daphnia dentifera, a local species. And I'll be separating them out to these 4 main parasite treatments. So we'll have our Mech, our MICG brood, and then a control group. And something really unique about this system is that the infectious spore slurries that we prepare from these parasites are actually typically done through blending the whole animal, as the parasite spores cannot be cultivated outside of the host. And so that leads the question is, oh no, we're going to accidentally contaminate your treatment groups with accidental donor microbiota. So I have these parasite-dependent protocols set in place to filter and isolate the spores free from their initial the host body parts and from bacteria. I will be happy to talk about this in detail later, but this could take up my entire presentation if I got too into it right now. And so once these guys are dosed with their different treatments, I need to set my time scale up, right, to track my microbiome changes in my species evenness, richness, and again, that community turnover. So therefore, I will randomly select and sacrifice 16 individuals at each time point. Now, my first one is for initial exposure. What happens when they just come into contact with that spore and it sits around, right? So this will be done at 24 hours across all of my, I lost laser privileges. Across all of my treatments. And then my final time scale will be at the maximum infection time point. And now because these guys are so different in their virulence and their life strategies, this will be a little bit different depending on the parasite. So for Mechanocobia, this will be after 10 days. And I will also sacrifice 16 control individuals to match it. And then for my McGene brood, similar virulence, I will sacrifice these guys at a 28-day post initial exposure. And then that'll be the final round of sacrificing for control as well. Here.
The whole thing died.
I don't know. So once I have all these samples collected and sacrificed, I will be using something called 16S RNA sequencing, which I'm sure most of you have heard of by this point. Now, what this does is it targets a highly conserved gene region of bacteria, the 16S RNA. And so what it does is it allows us to compare composition and run diversity metrics. So it tells us who's there in what abundance, right? Yeah, I think this is dead.
I have to use the mouse.
Yeah.
Sorry.
Okay, sorry guys. So there are a couple of issues with 16S RNA sequencing. However, most studies on Daphnia microbiomes have used this because it's pretty fast, it's cheap, and there's these established pipelines for analyzing your data through this. However, once again, it neither is able to differentiate your different bacteria at the strain or species level, and it does not provide information about functional capabilities. So what are they actually capable of doing, right? So I'm actually using my 16S RNA sequencing as a way to pre-screen interesting treatment groups that might have a cause for me to process them through whole genome shotgun sequencing. So what this will do is show me the entire potential suite of genes present within the microbiome. I will do this to determine how the microbiome structure and gene expression changes across different parasite treatments and across different sacrifice time points. And combine this with RNA-seq. And now what RNA-seq will do will actually track the presence of RNA to show gene expression during my different conditions. And from there, I'll be able to do differential gene abundance to compare the expression levels between my treatments. So we have another pretty important question. So this chapter 1 will be split off into 2 chapters, two experiments actually. So the goal of this one is to be able to differentiate between the direct effects of parasite virulence and then look at the microbiome dysbiosis itself and its role in causing a reduction in host fitness. So I wish I had my laser now. I will set up germ-free Daphnia animals. So I'll collect a bunch of my adult mid-37 clone lines. And what I'll do is very gently, not so much, harvest the embryos out of their brood chambers, stick them in this petri dish while I hit with an antimicrobial wash, clean that off using an autoclave lake water, allow them to develop for 2 days, and then, okay, yes, wait. And then I'll inoculate them with microbiomes that are representative of each of these original treatments in the absence of spores. So it's like that inverse of filtration spore isolation I showed you, where instead I'm trying to remove the spores So I'm inoculating these treatment groups with the microbiome itself after these hosts have been maximally exposed to their parasites. While this experiment is running, I'll also be taking lab table data. And this is to track offspring production for fecundity counts, determine mortality rates, and looking at lifespan changes in order to kind of correlate this with these dysbiosis microbiomes, right? And so by day 30, all of my remaining animals are actually going to be sacrificed for a DNA extraction, and then later on my 16S approach. And I think I already said this, but once again, this is just to see if it correlates with one another and to see if these are both interacting at once to really shut down these hosts when they're under stress. So let's kind of revisit my predictions. Once again, I think that the host fitness will decrease dependent on microbiome inoculation. I expect that those with these more virulent or disruptive parasites will have a more destabilized microbiome and thus leading to a greater reduction in host fitness, more dramatic. I also expect that because they're going to be kept in azemic conditions outside of that starting inoculum, we'll still see a change in the microbiome composition, but this will be a more abundance based as some species are able to better survive under these conditions. And again, across all treatments, I expect lower alpha diversity, and I expect that to parallel with lower functional diversity as well. As they start to lose symbionts, they lose the potential to have these adaptive responses to other stressors or other potential. Okay. And so this should change once again in our microbiome. The symbionts within us don't stay the same. They're all always going to be mutualist or parasitic. So I do expect shifts in the symbiotic relationships between the host and its microbiome, and that the infected individuals will have a distinct functional profile from uninfected individuals. So let's take a step back from the lab and remember where Daphnia come from, the outside world, right? And so we have this pretty stressful scenario here. You look at that top right corner, there's this cyanobacteria bloom that's causing some harm to local organisms. You can see here some parasite spores floating around in the column. Somehow marine plants showed up in my lake, but we're going to ignore that right now. And then you have fish, and then once again, fish are predators, right? And so now species, especially Daphnia, face simultaneous pressures with this temporal and spatial overlap of these co-stressors all occurring at the same time. In some cases, this overlap is determined by environmental variables, such as light availability or weather fluctuations. And that stressors that are abiotic can sometimes be more predictable in determining the outcome of microbiome composition. These abiotic stressors are able to create environmental conditions with strict parameters dictating who can live and survive there, reducing microbiome community complexity and making outcomes more uniform across similar environments with the same pressures, right? On the other hand, our biotic stressors are context dependent. You also have these species interacting with one another to create these different types of outcomes. So let's talk about some biotic stressors. So we have stressors that are associated with anthropogenic interference and climate change. So this is some example of cyanobacteria blooms in local lakes near University of Michigan. So these guys are increasingly frequent due to nutrient runoff and increasing temperature, right? And this is horrible for everybody else involved in this lake because cyanobacteria happen to be excellent competitors. They produce toxins that are detrimental to the macroorganisms present, but also have antimicrobial effects. They are able to uptake resources much quicker than their competitors, and they can also create these dead zones which are hypoxic, or they can block off light from reaching lower levels within the water column. So what I'm saying here is that the competition induced by these cyanobacteria blooms will, of course, cause reassemblage of the aquatic microbial community, and therefore our Daphnia microbiome. So this leads me to my second question, that as we have higher prevalence of cyanobacteria blooms taking over from this healthy nutrition green algae, how do these resource-driven shifts in the gut microbiome alter host susceptibility to infection? So do these combined exposures have a synergistic impact on gut microbiome composition? And does the gut microbiome play a protective or detrimental role under dual structure conditions? So a little bit of background here. Daphnia acquire their symbionts through horizontal transmission. There's no evidence of vertical transmission going on here. So Daphnia happen to be suspension feeders that intake their food and accidental microbes and parasite spores through filtration. And their filtering rate is dependent on the food availability within the water column. So they can kind of slow down and speed up their filtering. This subsequently facilitates the uptake of potential gut symbionts, both mutualistic and parasitic, as I mentioned before, via horizontal transmission. Whoop. Okay. So we do have some more evidence from previous research done that shows if you provide different inoculums, if you stick them in different environmental types, your gut microbiome composition is going to be pretty reflective of where they are and who's present as a potential uptake in a symbiont. However, it remains unclear whether host-associated microbial communities indirectly influence outcomes of infection that follow cyanobacteria blooms. So I'm going to combine these two stressors, my cyanobacteria and Mechdicobia infection, to investigate whether shifts in research type alter host parasite interactions through microbial turnover in the gut community. So to do this, I have chosen the fungal parasite Mech. Right? We know that Mech enters the gut via consumption. We know that it directly interacts with the gut for infection, and really importantly, it has low genotype specific, I can't pronounce this right now. It's not so specific over the type of genotype in order to have a successful infection, is what I'm trying to say. It's a very mechanical infection progress. And so this kind of leads up the potential for instead the microbiome being able to confer some sort of role here, right? So we can kind of look at these guys. Now, under natural conditions, there's more stressors, but in my lab setup, they'll be under selective pressures from both my cyanobacteria bloom and from parasites. Now, previous work conducted in the Daphnia lab has shown that these two stressors do interact with each other. So cyanobacteria can actually deactivate important host proteases in the gut and prevents the spores from being released from their ascus and therefore being able to have a successful infection. And this work was done by Crystal Sanchez. So let's talk about some predictions really quickly. Cyanobacteria exposed treatment groups will probably differ significantly from other. So as I previously kind of showed you guys, cyanobacteria can the microbiome can mediate tolerance to the cyanobacteria. However, it's not known to do this for parasitic stressors. So I expect that in the microbiome and in the outside water environment, we will see a decrease in number of microbial species. Also, stressing events also normally occur and is marked by limited or lowered biomass throughout. And so I should see this in the microbiome, and I'll do this using qPCR to get a quantification, and then a change in abundances of species throughout. And more so, I expect the cyanobacteria inoculum to have these huge effects, right? Further, when we combine these two stressors at once, they could interact in different ways, intensifying dysbiosis. So in groups that have been exposed to both cyanobacteria and the parasites, I expect to see this super dramatic decrease in host fitness, or perhaps there will be no interaction between those two whatsoever, no additive effect, and they both occur independent from one another and not mediated by the microbiome. So for methods, and bear with me here, I'm going to have 3 starting genotypes. And now these genotypes will be picked in varying levels based on mesh susceptibility, as limited as they may be. We do have them in lab. And so these animals will age up for 10 days before being randomly assigned. And this is to ensure that they're mature and their guts are of full size, right, for visual colonization. Then they'll be randomly sorted into 2 exposure groups. So these will be exposed to cyanobacteria, or not exposed and fed their normal healthy green alga. And what the purpose of these 2 treatment groups is that they're going to be sacrificed and prepared as microbiome inoculates. So these are actually my donor animals. So don't get attached to them, right? So while this is all running, I'm going to go back to that xenate methods and produce a ton of germ-free eggs, which are once again placed into these individual capped falcon tubes and inoculated with these donor microbiota that are either cyanobacteria exposed or not exposed. And they'll be allowed to develop for 7 days to allow this to establish and stabilize. So, I'm sorry if this gets a little convoluted here. So my donors will then be spread out into all these treatment groups where I'll be covering all 3 genotypes. There'll be 20 animals per of these final groups. And so once they have this established microbiome based on the inoculum I gave them, I will separate them out based on ones that will receive parasitic slurry and ones that will not be exposed to my parasite. And now at week 2, so if we remember, mesh is highly virulent, I will be randomly selecting and sacrificing a handful of animals from each of the treatment groups to preserve for microbiome analysis, such as qPCR for biomass, and then my metagenomics. So now I was kind of hit for my chapter 3. We are now informed from these lab experiments about whether or not these guys are directly interacting with the microbiome and if the microbiome is playing a role between these different stressors. So how do these cyanobacteria blooms and parasites interact to shape the community assemblage of host-associated microbiomes over time? So this is a really fun paper done by a research group up in Belgium, where they look at how the host genotype dependent gut microbiome is actually able to provide tolerance to toxic cyanobacteria. And so here, I'm going to come over here and point. So this is an example of 4 genotype lines or clonal lines that they established, and they looked at the survival probability over time when they were exposed to cyanobacteria. So this S actually stands for susceptible. So these are the ones sensitive to your cyanobacteria. The T is tolerant. So those that are able to resist, right? And so we expect those tolerant genotypes are better able to survive. And what they did were these reciprocal transplants. Right, and so if we look over here where the recipients receive the same microbiome, we don't see any change, but this is the important graph here. And now what this tells us is that when your susceptible genotypes are inoculated with microbiomes from those tolerant genotypes, they're actually having higher survival probability over time. And then the inverse is also true, where these sensitive genotypes, once inoculated with microbiomes, yeah, just that. So these tolerant microbiome genotypes, when inoculated with sensitive microbiomes, have a much lower fitness and are less likely to survive over time. Okay, and one final thing, they were able to kind of quantify some of the overabundance of certain microbial groups within these tolerant microbiomes. And these included higher abundance of Cyanobacteria, which are able to degrade microcystic cells and release that dissolved organic carbon back into the water column. Or there were other groups that were able to directly degrade the toxin itself, not necessarily the cyanobacterial cell. So Meg didn't love this picture because it's not a real boat, but I couldn't find a canoe, I'm sorry. So this is me on the field. As we can see, we have all these stressors going on, and I have my little plankton net, and this is because I will be doing a field-based approach to investigate my Daphnia dentifera populations across several lakes in Michigan. Now, I'll have a set of 3 lakes that do undergo naturally occurring cyanobacteria blooms, and then 3 lakes that do not. And something important about these lakes is that they're all going to be shallow. And what this ensures is that there's no deep water refuge for my Daphnia to migrate up and down and hide the cyanobacteria. So if it's there, they have to deal with it. I will be collecting these guys using a Wisconsin style plankton net, and I will be sampling throughout our field season, which is from July to November on an every other week time frame. And this is to allow me to do a time series analysis of the fluctuations in the gut microbiome assemblages. So once again, I'll be doing plankton toes. I'll also be taking water samples across the water column. And this is so I can also look at whether or not who's present is also changing due to these stressors. And if there's direct transmission between the aquatic environment and my microbiome, my host-associated microbiomes. Before transport back to the laboratory, I will be fixing my samples in ethanol in order to preserve the microbiome state as it was during sampling time. And this is for later DNA extractions. Yeah, and now I will be using that same approach I used in the first one. However, this will just be 60 mass, and I'll tell you why, because it's a little bit exciting in a second. Now, a portion of each of my Daphnia samples will also be preserved once again. This is to allow me to hit them with some primers and probes, using dPCR to see if they are infected or there's latent infections that might also interact with the gut microbiome. And of course, I'll be taking some metadata about what's going on, what is the light availability looking like, how's the weather, because all of these environmental variables play a pretty important role as well in shaping microbial communities. So what I'm hoping to do here is something called microbial co-occurrence networks. And so what this looks at is the presence or absence of certain co-occurring microbes with one another. And it's able to define how they can interact potentially. So if you're using 16S, this is more of an inference based where you can look at what their potential function is, but once again, there's no causal link between the two. So we look at this side. This is what happens when you have a destabilized microbiome hits with a stressor, right? So some important ideas here are that communities with more positive cohesion are more unstable. And now here, positive stands for like these niche overlaps, right? You're competing with one another, you're having these antagonistic interactions. Net cohesion is the opposite, where you're facilitating the growth of each other. And in the microbial sense, it's normally through cross-feeding of secondary metabolites where you're dependent on each other. And so you're able to create these modules of interacting nodes. And the idea here is if you have a higher modularity, that means you're kind of set together, and this can be based off of functional types. So you can create these functional groups more closely associated with each other. And you have a ton of interactions going between other species in your module, the limited interactions with other modules. And so the idea here is if you're highly module, then you'll have one module, and as you're all similar in function to each other, that might be more sensitive to your particular stressor. And so this disturbance will occur, heavily impact this module, but because of limited links to other ones, you're not going to see this resounding effect across the entire network as much. On the other hand, if you're all highly interconnected with one another, that disturbance is going to have cascading effects across the entire way, right? So what I'm thinking here is that my disturbance is going to be cyanobacteria. So I'm thinking that cyanobacteria as my biotic stressor is going to lead to greater destabilization within the microbial network. And once again, these are indicated by a decrease in modularity and an increase in your positive association to negative association ratio. This doesn't bode so well for my Daphnia because once again, once they're impacted and they might be destabilized, what happens when you have that immediate second biotic stressor coming in and you're already weakened from the prior effect? So it's important to uncover how these communities fluctuate over time and under stress. So as I kind of repeated what I just said, I expect these guys to suffer way more later on down the line. And what will probably happen is that my cyanobacteria blooms will lead to a reduction in microbial diversity and thus a reduction in functional diversity. And this is important because if you have a higher diversity, you have more likelihood of having some present species in there that are able to confer the special ability not encoded in other species. And similarly, I expect my parasite prevalence to also cause the shift in microbiome composition. Now, when these both overlap or occur right near each other, I expect that these will be increasingly disrupted. So a cascade of effects resulting in a destabilized network, not only just the host-associated microbiomes, but I also expect to see a similar disassociation within the aquatic community in the water column. So in conclusion, I hope I convinced you that Daphnia are pretty important within food webs, that they experience extreme stress all the time, that Daphnia also rely on their microbiomes to be able to survive, and that my research will hopefully uncover how common stressors alter the microbiome and the resulting implications on the host's impact with resources in the greater environments. So, yeah. I'd like to say thank you to the Daphnia Lab in particular. I'm missing a couple of people, but we're notorious for being hard to get for a group photo. Special shout outs to Merchant Juba, who helped me a lot, and to my Mia Robbins, who's currently helping me set up these experiments in the lab. And I guess now.
Thank you, that was awesome and so thorough. I guess my question is like a little bit about methodology, but it's just a broader question of seems like the parasites are like micro eukaryotes, like fungi and oomycetes. So I was wondering if that's part of the microbiome, and if 16S, if you expect all the microbiome to be bacteria,
Yeah.
or if there'll be other fungi in there that are also part of the piece of microbiome.
No, great question. And so microbiome is not limited just to your bacterial symbionts. It includes viruses, parasites, microfungi as well. And so I'm kind of limiting my scope. I'm looking at the bacterial associations, but with the metagenomic parts, I will be looking across groups.
Awesome talk. I just was curious, in your first chapter, you were talking about these 3 different parasites, but it seemed like your experiment is them set up mostly in isolation. And I just was curious, in lake systems, do you see Daphnia infected with multiple parasites?
Okay, I almost threw up this really fun graph that shows parasite prevalence, like all the different parasites within the lake. And so, yes, you can see this time lag between epidemics. You can see parasites occurring at the same time, like outbreaks occurring similar to each other. Like within the lake, there's so many different parasites. I'm trying to focus down on a couple, especially ones that our lab does a lot of like inside work on, like bench work with. But yeah, you do see a ton of overlap.
Do you have a hypothesis if you had 2 parasites and 1 Daphnia? Do you think you'd see similar outcomes, or do you think they'd interact synergistically in some way?
Okay, fun question. So our postdoc is actually looking at I think this question's better for him or Chin, but I'll answer for you. And so what he's doing is he's looking at co-infections of MIC-G and METCH. And so originally, when you're looking at the individual scale, it can almost seem like MIC-G is preventing a successful infection by METCH by kind of blocking them from being able to pierce through the gut. However, what you see on population level scales is that MIC-G is actually way worse when you have both infections occurring at the same time, completely reducing the fitness of the offspring with something called transgenerational virulence.
So cool, thank you.
How about if we could stand down?
No, I didn't want it.
It's a cute and great system. I'm curious about the RNA sequence of your first chapter, because it's a lot of information when we make like a transcription. So I'm curious if you could take advantage of like the genomes published for Daphnia to choose some candidate genes for.
Yeah, exactly. So I'm hoping I'm kind of going to make some educated guesses about what I think is occurring, what might be overly expressed or maybe even downregulated. And this will probably be in certain like secondary metabolite production, or once again in some sort of process being able to degrade, especially the chitin of some of my fungal parasites. So I can be targeted toward that. There is a collaborator who has done metagenomic assemble genome work before, and so I'll be able to use that database.
That was a really nice talk, thank you. So for your first chapter, I think you were sampling after like different amounts of time for different parasites.
My first, here, let me find it. This one?
Yes, yeah, here. And so do you think the duration of infection will affect the strength of the response in the microbiome at all?
Yeah, 100%, which is why I'm doing it across time points. So something I didn't mention here is that MIC-G and brood are also more cyclical. And so they can hit that maximum parasite burden, but then also drop back down to a lower burden before going throughout the host's lifespan. And so I'm hoping that the time point I've selected will kind of pinpoint them at that range. I've included some extra numbers in case not every one of my hosts wants to cooperate with me. So that will help you buffer, but I do expect, that the time span, so if I compare my maximum infection, compared to my initial exposure, you should see a greater difference in magnitude of compositional change. And again, that's kind of one of the reasons why I'm equally sacrificing my controls at points that match each sacrifice point, so I'll be able to compare that difference between the control across my treatment groups, right?
Okay, cool, thank you.
That was awesome, thank you. Can you remind me how many, let's say, prokaryotic players there are in the microbiome, just to not piss off the fungi people?
On Daphnia specific microbiome? A ton, so let me pull back up. So we are using 16S, once again, we're not able to discriminate between different parasites at the strain or species level, but we look at the phylums. We have a pretty wide variety here. I think this is like almost 30, maybe, or less, probably less, I can't count right now. And this is from the wild, and so we expect that when you are using lab animals, you're gonna have this reduced microbial diversity present, as you don't have all sorts of environmental inputs of potential symbionts. They're kind of limited to like, oh no, somebody stuck their finger in the water. So you'll have that in there, but.
And is this distribution distinct from like the background water?
Yes, and so there is host selective uptake from your environmental source, and so you can see it's diverged from that aquatic collection microbes.
Really good. At what day was this sample done? I'm just curious.
What day?
Yeah.
I'm not sure in the actual day. I just know it's during the field season, so sometime in late summer, early fall. Hi.
No, I was thinking more about like, since the time since infection. I
Yeah,
guess.
well, that's why my chapter 3, I'll be doing like a time series selection, and so it'll be across the field season, different.
Yeah, one right here. Yeah, nice job. Regarding the chapter 3, I'm just curious about the time series, so you said every other week, is that right?
Yeah,
So 2 questions, I guess. One is that enough time to get the spatial dynamics over, is this one field season?
it'll be in one field season.
So is that enough time to do that in terms of like frequency, and then also is that enough? I'm not sure I understand the analysis, but like you talk about time series, do you have enough power with that many data points to actually
Yeah,
address that?
that's a great question, because that's something I'm actively like thinking about right now. So the reason the original time selection is picked, because that is how we sample our lakes in our established field season. So the infrastructure is already set up for me to go out and do the sampling that way, which is the most convenient method. I have thought about doing it every week or maybe even like twice a week, to get higher, like better resolution on the fluctuations in the composition. So yeah, I would probably, when I hit chapter 3, break it down into getting like more frequent sample points.
Yeah, you could even like, just even for a chunk, you can't do it frequent the whole time,
Yeah.
just like do a chunk and then you can kind of zoom in and then like scale out and you can even.
Yeah.
you can-
If I like pinpointed like the times when I do have that higher Cyanobacteria exposure in this collection of 3 lakes, that would be my targeted time. If I broke it down inside the entire field season.
Cool. Thanks, Joe. Anybody want a cue? I'll throw it.
If that's it, then let's give Riley another round of applause.